Well, good afternoon. This is Bio 212, North Community College. We're examining Chapter 24, and after some feedback from people who have looked at the previous sets of slides, there's two things. The first of which is they've asked for some of these to be a little bit longer. Let's see how that goes. The reason why we actually have shorter time is it seems like anything above 15 minutes, people tend to lose their ability to focus. But I'm going to try and break these sets of slides up into about 20 minutes today and see how this goes. So Chapter 24 is what is the digestive system? And we want to begin with the fact that there are two groups of organs that compose the digestive system. And that's the gastrointestinal tract. And of course, everything from the tips of your lips to the bottom of your anus is what we're talking about. And the alimentary canal, which is what I just described for you, is the mouth, most of the pharynx, the esophagus, the stomach, small intestine, and large intestines. Okay, that's, if you think about what I have drawn inside class, that's that tube inside of a tube, right? The accessory structures, teeth, tongue, salivary glands, liver, gallbladder, and pancreas. Specifically, say the liver, the gallbladder, and the pancreas. When we're looking at how is it we can take something that's solid. In class, I used the example of, what was it, chimichanga, right? Let me just put in red here, something red, and that red is going to be food. Okay, and that food is going to pass its way through all of these structures, right? Through the alimentary canal. And as it's going through, materials are going to need to be secreted into this so we can actually, at the end of the day, turn them into monomers of whatever they once were as best we can. So really, in essence, what we're doing is if this was a piece of chimichanga or a piece of steak or a donut or whatever, by the time it actually gets through to our small intestine and exits the small intestine to enter the large intestine, it's been broken to its potentially smallest pieces. Okay, it doesn't always happen, but for the most part, that's our goal. That's what you want to be able to figure out. Okay, get rid of that. That over here. And what you can see in this figure from your textbook is these are the organs. And, this is no different than what I drew inside class for you. Come over here. I'll even make a more simplified version. Okay, change the color. Okay, we're just going to go like that. Boom. Stomach. Small intestine, twink, 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 twink. Large intestine, twink, 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 twink. Right? And so that makes this the esophagus. And if we really want to be smart about this, we want to make sure we put in a mouth so that materials can actually be ingested into the system. Right? And as you're going along, remember what I asked you to do inside class is sort of figure out where chemical and mechanical events are going to occur, right? Okay, and what's going to happen, of course, is as we make these monomers of these macromolecules, you need to remember that all tissues, all food materials that you're ingesting are made up of protein, P for protein. What is the other? Carbohydrate. Okay, so if we have proteins, we have carbohydrates. Remember, proteins are made up of amino acids. Carbohydrates, lipids. Okay, nucleic acids, Na. Okay, that does not mean not applicable. Okay, once you get this idea, now we're going to pass whatever that solid structure is, the monomers of each of these. So remember, that means there are 20 different types of amino acids that could now be absorbed into our body to be processed someplace. Carbohydrates come in many different forms, lipids many different forms. Nucleic acids, for the purposes of this class, we worry only about, say, RNA and DNA. Okay, cool. So there are 6 basic functions to the digestive system. We have to ingest materials. Materials need to be secreted into the lumen of that alimentary canal. And then we want to mix and propulse. Okay, and that propulsion, of course, is going to lead ultimately to the anus to remove the material from the body, which is the elimination of feces down here. Digestion comes in those two forms, the mechanical and the chemical digestion. And the major mechanism is hydrolysis. Okay, once we've broken things down as much as we can, we will now collect things into blood or lymph. Okay, let's do this. Okay, because some materials will go into your blood and some materials will go into your lymph, and part of the work you're doing is figuring out what ends up where. We need to think about how that alimentary canal is actually constructed. The wall of the GI tract, okay, has a mucosa, which is the inner lining, and it's for protection, secretion, and absorption. Deep to it, this is how we know we're inside the GI tract, is made up of a lamina propria, which is the connective tissue with blood and lymphatic vessels, okay, as well as mucosal-associated lymphatic tissue. It has its own muscularis. Okay, so that's significant. It means that there can be some changes that are independent of the 2 or 3 muscle layers that will be deep to this. It has a submucosa. Okay, and that submucosa has more connective tissue inside of it, as well as usually longitudinal and circular muscles, but with the exception of the stomach, and contains many blood and lymphatic vessels. Okay, and of course that should just make sense when we're thinking about the fact that we look at the muscularis. Parts of the body are arranged for specific functions, okay? It's voluntary skeletal muscle found inside the mouth, pharynx, and upper 2/3 of the esophagus, as well as your anal sphincter, okay? So that means, in general, you can control what actually enters and exits your body, right? And then it's involuntary everywhere else, where you're going to arrange inner circular fibers and outer longitudinal fibers that will be controlled by this thing right here. Okay, let's go bold. Okay, the myenteric plexus. You'll see how that happens in a minute. We have to hold it all in place, which means we actually put a visceral peritoneum around it, and the visceral peritoneum is what we call a serosa, unless we're inside the esophagus or paying attention to this esophagus. It has an adventitia. Let's take a look at what that looks like. Here's the inner lumen of the mucosa. Okay, meaning these are mucosal cells, inner space here. Materials you've ingested are here. Glands that are supporting secretions that move into this space, okay, will divert materials into here. And in yellow are the actual nervous system innervations to control not only that muscularis mucosa, okay, but also the muscularis here. Okay. In general, or in total, I guess I would say, the neural innervation inside the gastrointestinal tract is called the enteric nervous system. Brains of the gut is what I put here, right? And it has the myenteric plexus, which is the GI tract motility, and then it has submucosal plexuses, which are controlling secretions. How does the stomach know when to secrete X, Y, and Z, meaning mucus or hydrochloric acid, okay? It's essentially part of the autonomic nervous system, where the parasympathetic nervous system stimulates secretion by activating the ENS. Sympathetic stimulation decreases secretions and inhibits the ENS, which makes sense, right? This is, you actually have time to sit back and relax and enjoy the meal you're eating. Parasympathetic nervous system is activated. You're running for your life. You're not thinking about what you're eating. You're trying to survive. What does this actually look like? Okay, here's our ANS regulating the myenteric plexus, which is in turn going to regulate the submucosa. And ultimately, we will control through the myenteric plexus the longitudinal and circular muscles, as well as the mucosal epithelia. And they are actually sensing what's actually passing by them. The peritoneum, okay, the largest serous membrane inside the body. So this is the largest serous membrane inside the body, and from AMP1, you know it's divided into the parietal, which lines the walls of the cavity, and viscera is going to cover some of the organs, and it's also called the serosa. We've discussed this many times. What I want to make sure you understand is how Mother Nature separates all of these inside of your body. So if that really simple cartoon I've been drawing for you is an oversimplification where you actually pack all of these tissues in there by holding on by what we're going to collectively end up calling the mesentery to hold this all in place. Once you understand how it's all packed inside these cavities, or the cavity, abdominal pelvic cavity, we need to turn now our attention to the mouth, where you actually have a series of teeth, but also a series of glands. And Teeth are pretty simple to understand. No big deal. They're hard. They break things apart. Big deal. They're awesome when they're designed, and they're really cool to think about in terms of engineering, but they're pretty straightforward. Apply a force to a softer material, the enamel of your teeth cuts through it. Salivary glands, though, however, ordinarily they just keep the mouth clean, believe it or not, and when food actually enters the mouth, though, the secretions are increased to lubricate, dissolve, and actually begin chemical digestion. And what you need to make sure you understand is there's a parotid, submandibular, and sublingual gland. So I can point these out to you. Okay, so here's the, where are they? Sublingual. Nope. Opening of the duct for the submandibular gland. So notice this is your lingual frenulum. This is underneath your tongue. So your tongue has been reflected, and literally liquid comes out of here, right? And if you just start thinking about food, that should help you actually feel the difference. Here are the three glands in focus for you. The parotid, notice on the side of your face. Take your fingers, push into your jaw. You'll feel them being hit. Sublingual, okay, are actually right underneath the tongue, okay, and this right here. And then submandibular, okay, beneath the mandible, okay. Obviously, they're making materials. For the most part, we want to ensure that you understand that you have salivary amylase and lingual lipase inside your vocabulary. There's also ions, gases, uric acid, mucus, immunoglobulin A, as well as, where was it? What did I skip over? Uric acid. Sorry about that. But it's mostly water. It's controlled by the parasympathetic nervous system. Okay, and anytime you've ever been nervous, your sympathetic stimulation shuts that off. The tongue itself is also a digestive organ only because it's a muscular structure that shapes the masses of food and forces it to the back of your throat for actual swallowing itself. And these, the lingual glands here, are going to supply that lipase I was just talking about. All right, here are the teeth. There are three major regions. Each tooth has a crown, a root, and a neck, but who cares? Okay, how it works. Mechanical digestion inside the mouth will form the bolus. Chemical digestion, where we actually use the salivary amylase, is 3 times in like 3 seconds. I've said this to you, right? So salivary amylase, lingual lipase, okay? Because when we are ingesting materials, or it's say absorbing materials inside our small intestine, they have to be monosaccharides. Salivary amylase is going to begin the process, okay? It's going to continue until it's inactivated inside your stomach, and lingual lipase begins to break down triglycerides. As food passes from the mouth into the pharynx, you should understand the nasopharynx functions only in respiration, inhalation, and exhalation of gases. Oropharynx is the digestion and respiration functions, and laryngopharynx, the digestive and respiratory functions as well. Okay, so you can break these apart further, okay, when you're thinking about how they're going to function. Let's think about the esophagus, though. Materials are going to travel into this portion where there's no enzymes produced and no absorption occurs. So it's purely for transport, okay? And it has a mucosa to protect against wear and tear, and it sits on top of a submucosa. The muscularis, meaning to propel materials through, you'll see the photomicrograph in a moment. The superior 1/3 is skeletal muscle, translated into this is voluntary. Middle 1/3 is skeletal and smooth muscle, partially voluntary, partially involuntary. The inferior 1/3 is smooth muscle, which means we have no regulation over it all. The lower esophageal sphincter regulates movement into the stomach, and it's attached to the surroundings, meaning the back of the trachea, using an adventitia. Here's what this looks like. It's the mucosa itself is non-keratinized, stratified squamous epithelia. It sits on top of a lamina propria, and here's where you actually see the lamina propria and the muscularis mucosa here. But down here you have the circular layer and the longitudinal layer that sit on top of that adventitia. Big word for swallowing materials, deglutition, fancy, okay, the act of swallowing, facilitated by secretions of saliva and mucus. As I said earlier, voluntary, the bolus is passed into the oropharynx. Pharyngeal is involuntary, passage of pharynx into esophagus, and esophageal is involuntary passage of materials from the esophagus to the stomach. It is the mechanism of peristalsis. Let's go bold, underline. That actually pushes materials forward. Okay. Here you can actually see the bolus. Let's say chimichanga, just for the fun of it, right? Travels through circular muscles and longitudinal muscles. So circular muscles contract, pinching it. Longitudinal muscles contract, actually having it move forward. That's because this is a mucosal tissue and things don't get stuck. Think about the last time something got stuck on the way to your stomach. Okay. The stomach actually has four main regions: cardia, fundus, body, and pylorus. It's a mixing and holding reservoir. Remember, at the end of the function here, we have to have gotten materials into a liquid state. So to do that, we have a mucosa, which has gastric glands that open up into gastric pits. And you'll see in the next slide the same cells we looked at inside class, where you actually have mucous neck cells, which secrete mucus, parietal cells, which are going to make intrinsic factor and hydrochloric acid. Oops, that's not what I wanted to do there. Let's go like that. Boink. Like that. There you go, like this. All right, and hydrochloric acid, as well as chief cells. Those chief cells make pepsinogen and gastric lipase. And then there's also another population of cells that are called G cells. These are endocrine cells, which means now the stomach is an endocrine gland, and it secretes the hormone gastrin. Okay, so obviously do that and that as well. Okay, so here's the cells I wanted to show you. Remember, these are the same thing you see inside your textbook. Here are the mucosal cells. Mucosal neck cells makes mucus as well. And here are those parietal cells. They're actually going to make hydrochloric acid. Here are those chief cells that secrete pepsinogen and gastric lipase. And here are the G cells way down here. They actually make the hormone gastrin. So how does it all work? Material gets inside your stomach. You actually have mixing, gentle peristaltic movements to create chyme. So that's the hydrochloric acid tearing everything apart. Okay? And what's going to happen is the acid gastric juice as well. And what I mean by that is the salivary amylase that came into your stomach is going to be inactivated. Acidic gastric juices are going to activate the lingual lipase. Okay? And what's going to happen is now you're going with the, what's going to happen is you're now going to kill many of the microbes and denature proteins. Okay? Pepsin is going to be secreted by chief cells, and that's going to happen there is you're going to start to digest proteins. But to get pepsin, we first have to secrete pepsinogen, okay, which will then be converted into pepsin. And gastric lipase takes triglycerides and breaks them down into smaller monoglycerides. But this is an ongoing event inside the body and shouldn't be thought of as the only place this occurs. There is a small amount of absorption that happens inside your stomach. Definitely alcohols, aspirins, or aspirin, and some short-chain fatty acids. That's where the folklore comes from. If you want to keep from getting drunk, you have a little bit of olive oil. How does your digestive process work next is what we want to do is we want to pause and say that there's a supporting structure, the pancreas, which is going to lie deep to the stomach. Remember, it's covered in connective tissue, and it's going to make materials. And what's going to be making, of course, are pancreatic juices, which are a mixture of fluid and digestive enzymes. And then inside that fluid is going to be bicarbonate. Let's make sure that's inside there. HCO3 minus, right? Okay, so, and the remaining, of course, are glucagon, insulin, somatostatin, and pancreatic polypeptide. That's chapter 18. This is chapter 24. Okay, so what's it doing? Well, you make it, and it's going to be collected and travel through this pancreatic duct. And in green here, you're going to see materials. Whoa, what just happened? Okay, materials are being made here inside the liver, which we haven't gotten to yet, are now coming down as well, and they're mixing here. Okay, at the hepatic pancreatic or hepatopancreatic ampulla, okay, also called the sphincter of Oddi. Okay, and whatever's now come into the duodenum from the pyloric sphincter, you're now going to mix with that chyme, and you're going to offset the pH. What's inside of it? You make about 1,500 mils of this stuff daily, mostly buffers for the stomach chyme. That's the purpose of the sodium bicarbonate. And the enzymes are pancreatic amylases, proteolytic enzymes, and notice every single one of these is targeting a specific type of protein or connective tissue, which is made of a protein. Elastases, think about all the elastin you eat in terms of like the cartilages and just around blood vessels, I suppose. You have a proelastase. Carboxypeptidases are going to break off carboxy terminal peptide bonds. Chymotrypsins are working primarily on trypsin, and trypsin itself is secreted by as trypsinogen, and these are going to work on breaking down these specific proteins. We also have here, this thing is called pancreatic lipase, so now we can actually begin the third phase of lipid digestion. Okay, and finally we actually have nucleases, RNA and deoxyribonuclease, to break down RNA and DNA here. And that's a pretty good place to stop for right now. So I'm going to load this thing up and then I'm going to work on the next one. See you guys in a bit.